Good afternoon, and thank you for joining me as we talk about the cell cycle and its regulation mechanisms of control. To start, the cell cycle is a fundamental process by which a single cell grows, duplicates its genetic material, and divides into two daughter cells. It is essential for the development, growth, and maintenance of all living organisms, from single cell bacteria to complex multicellular organisms like humans. The cell cycle is tightly regulated and ensures that cells divide accurately and at an appropriate time, maintaining genetic stability and tissue homeostasis. The cell cycle is typically divided into four main phases: gap 1, synthesis, gap 2, and mitosis. The G1 phase, or the gap 1 phase, is a period of cell growth and preparation for DNA replication. During the S phase, the cell duplicates its DNA, ensuring that each daughter cell will receive an identical copies of the genome. The G2 or gap 2 phase is another growth phase where the cell prepares for mitosis by synthesizing necessary proteins and organelles. Finally, during the M phase, or mitosis, the cell undergoes mitosis where the duplicated chromosomes are evenly distributed between the two daughter cells, following by cytokinesis, the division of the cytoplasm. The transition between these phases is tightly controlled by a network of regulatory proteins, including cyclins and cyclin-dependent kinases, or CDKs. These molecules help to ensure that the cell cycle progresses in an orderly and timely manner. Checkpoints are also in place to monitor and respond to DNA damage, incomplete replication, or other cellular stresses, halting the cycle to allow for repair or triggering apoptosis if the damage is irreparable. Dysregulation of the cell cycle can lead to an uncontrolled cell division and is a hallmark of cancer. Understanding the mechanisms of cell cycle control is therefore crucial for developing targeted therapies for cancer and other proliferative disorders. This introduction sets the stage for exploring the intricate molecular machinery that governs the cell cycle and its regulation and its implication in health and disease. Interphase is our first step in the cell cycle. During this phase of the cell cycle, the cell prepares for division. It's the longest phase in the cell cycle, accounting for about 90% of the cycle's duration, and it's divided into three main subphases: gap 1 or G1, synthesis or S, and gap 2 or G2. The duration of one cell cycle can vary significantly depending on the type of cell and the organism. In rapidly dividing mammalian cells, such as those in the early embryo or certain cancer cells, the cell cycle typically takes about 24 hours. However, this duration can differ based on the cell type and the condition under which it is growing. The G1 phase usually lasts about 8 to 10 hours. During this phase, the cell grows in size, synthesizes proteins, and produces organelles. The cell prepares for DNA replication by accumulating the necessary enzymes and energy stores. However, the length of the G1 phase can vary considerably, especially in cells that are not actively dividing, where it may be extended or the cell may enter a quiescent state or G0 phase. The S phase, during which DNA replication occurs, typically lasts around 6 to 8 hours. The primary event of the S phase is the replication of the cell's DNA. Each chromosome is duplicated to produce two sister chromatids, which are identical copies connected by a central mirror. Along with DNA replication, the centrosomes, which play a crucial role in organizing the mitotic spindle during mitosis, are also duplicated. In the gap 2 phase, which is usually shorter, lasting around 4 to 6 hours, the cell continues to grow and prepares for mitosis. During G2, the cell continues to grow and produce proteins, especially those needed for mitosis. This phase is also a time for further cell growth and the completion of organelle replication. The cell performs a final check for DNA damage. Any errors or damage detected must be repaired before the cell can enter mitosis. The G1 checkpoint, the G1 checkpoint, also known as the restriction point or start point, is a critical control mechanism in the cell cycle. It occurs at the end of the G1 phase, just before the cell enters the S phase, where DNA replication occurs. At this point, the cell is irreversibly committed to cell division. The checkpoint ensures that the cell is ready to begin DNA synthesis and ultimately proceed to division. The cell checks for adequate cell size, nutrient availability, and DNA damage. The cell monitors its overall cytoplasmic volume relative to its DNA content. Cells need to maintain a certain ratio between their cytoplasmic mass and its DNA to ensure that after division, daughter cells will have sufficient cellular components to function properly. Growth factors bind to cell surface receptors, initiating a cascade of intracellular signals, mainly through the PI3K, AKT, and mTOR pathways. These signals promote cellular growth by increasing protein synthesis and inhibiting protein degradation. Adequate growth factor signaling indicates that the cell has reached the necessary size to proceed. Cells also have specific sensors that detect the presence of key nutrients, such as glucose, amino acids, and lipids. These sensors activate signaling pathways, particularly the mTOR pathway, which integrates signals related to nutrient availability. When nutrient levels, particularly ATP, are low, the cell activates AMP-activated protein kinases, or AMPKs. AMPKs inhibit anabolic processes that consume energy. This protein synthesis and promotes catabolic processes that generate energy. High AMPK activity suggests insufficient energy, which would halt the cell cycle progression. If the environment lacks sufficient nutrients, the cell may halt the cycle or enter the G0 phase, a resting state where it remains metabolically active but does not divide. The cell also inspects its DNA for damage. If DNA damage is detected, the cell will delay progression to the S phase to allow time for repair mechanisms to fix the damage. If the damage is too severe and cannot be repaired, the cell may undergo apoptosis, or a programmed cell death, to prevent the propagation of defective genetic material. Finally, the cell receives and processes external signals, such as growth factors that influence whether it should proceed with division. These signals can either promote or inhibit progression through the checkpoint. As we talk about the cell cycle, we'll talk about each checkpoint, starting with the G1 checkpoint. The G1 phase is regulated by a network of proteins, including cyclins, cyclin-dependent kinases, or CDKs, and tumor suppressors. Cyclins are proteins that regulate the activity of CDKs, which are enzymes that phosphorylate specific target proteins to drive the cell cycle forward. During the G1 phase, cyclin D binds to cyclin DK4 and 6, forming a complex that initiates the phosphorylation of the retinoblastoma protein, or RB protein. Phosphorylation of the RB protein releases E2F factors, a transcription factor that activates genes required for DNA synthesis, allowing the cell to transition from G1 phase to the S phase. Proteins like P53 and P21 play a crucial role in halting the cell cycle if DNA damage is detected. P53, often referred to as the guardian of the genome, activates the expression of P21, which inhibits the cyclin CDK complexes, preventing the cell from progressing through the G1 point. This pause allows time for DNA repair. During the S checkpoint, this is our next step in the cell cycle. It's located in the middle of the synthesis phase. It's also known as the intra-S phase checkpoint, and it's a crucial regulatory mechanism within the S phase of the cell cycle where DNA replication occurs. This checkpoint ensures that DNA is accurately and completely replicated before the cell progresses to the next stage of the cell cycle. It also serves to protect the integrity of the genome by responding to DNA damage or replication stress that may arise during DNA synthesis. The S checkpoint monitors the progression of DNA replication, ensuring that replication forks, the areas where DNA is being unwound and copied, are functioning properly. If any issues arise, such as stalled replication forks or incomplete replication, the checkpoint activates a response to stabilize the replication process and prevent the cell from moving forward prematurely. During the S phase, the DNA is vulnerable to damage from various sources, including environmental factors like UV radiation or internal factors like reactive oxygen species or ROS. The S checkpoint detects the damage and activates DNA repair pathways to correct any errors. If the checkpoint detects damaged RNA, it halts the progression of DNA to prevent the replication of damaged or incomplete DNA. This prevents the propagation of mutations that could lead to genomic instability and diseases such as cancer. The S checkpoint works closely with DNA repair mechanisms to ensure that any damage detected during replication is promptly repaired. This coordination is crucial for maintaining the accuracy and completeness of the genetic material. So as we move through the cell cycle, we'll talk about the checkpoints and the regulators of those checkpoints. For the S checkpoint, some of the key regulators are ATM, ATR, CHK1, P53, the BRAC1 and 2 genes, the MCM complex, and CDC25 proteins. Starting with ATR. ATR is a key protein kinase activated in response to replication stress, such as stalled replication forks or DNA damage. ATR phosphorylates and activates several downstream targets that help stabilize replication forks, initiate DNA repair, and prevent the cell from entering mitosis with damaged DNA. CHK1, or checkpoint kinase 1, is a downstream effector of ATR. When activated by ATR, CHK1 phosphorylates and inactivates CDC25A, a phosphatase that promotes the activation of CDK2, which is essential for DNA replication. By inhibiting CDC25A, checkpoint kinase 1, or CHK1, delays the cell cycle, allowing time for DNA repair and replication completion. CDC25A is a phosphatase that activates CDK2 by removing inhibitory phosphatases. CDK2, in complex with cyclin A, drives the progression of the synthesis phase. In response to DNA damage, checkpoint kinase 1 mediated phosphorylation of CDC25A targets it for degradation, thereby preventing CDK2 activation and halting DNA replication. The BRCA1 gene is a tumor suppressor gene involved in the repair of double-strand breaks in DNA. It plays a role in the synthesis checkpoint by helping to repair damaged DNA and stabilize the replication forks, ensuring that replication can proceed without errors. While P53 is more commonly associated with the GAP1 or G1 checkpoint, it can also be involved in the synthesis checkpoint, particularly in response to severe DNA damage. If damage is detected, P53 can induce cell cycle arrest, allowing time for DNA repair, or it can trigger apoptosis if the damage is irreparable. The S checkpoint is essential for maintaining the fidelity of DNA replication and ensuring genomic stability. By detecting and responding to replication stress or DNA damage, the S checkpoint prevents the accumulation of mutations, chromosomal aberrations, and other genetic defects that could lead to cancer and other diseases. A failure in the S checkpoint can result in the replication of damaged DNA, leading to genomic instability and the potential for tumor development. Therefore, the proper functioning of the S checkpoint is critical for the health and viability of cells. Moving on to the G2 checkpoint or GAP2 phase. During the GAP2 phase, During the GAP2 phase, we experience the G2 checkpoint, which is at the end of the phase right before the cell reaches mitosis. It is a crucial control mechanism in the cell cycle, which is a series of phases that a cell goes through as it prepares to divide and produce new cells. The G2 checkpoint occurs near the end of the G2 phase, just before the cell enters mitosis. The G2 checkpoint ensures that cells do not enter mitosis until they are fully prepared. This includes verifying that DNA replication in the synthesis phase has been completed accurately and that there are no DNA damages or other issues that could compromise cell division. During the G2 checkpoint, the cell assesses several factors, including DNA damage, replication errors, and cell size and energy. It checks for any DNA damage or errors that may have occurred during replication. It verifies that all DNA has been replicated properly. It ensures that the cells have grown to the appropriate size and has sufficient energy reserves. If the cell passes the G2 checkpoint, it proceeds to mitosis or the M phase. If issues are detected and cannot be repaired, the cell may undergo apoptosis to prevent the propagation of damaged DNA. The G2 checkpoint is a critical control mechanism in the cell cycle that ensures cells do not enter mitosis prematurely. The G2 checkpoint helps maintain genomic integrity and prevent the propagation of mutations. Some of the major regulators in the G2 phase, once again, are ATR and ATM, checkpoint kinase 1 and 2, cyclin B, and CDC25A and B, as well as some tumor suppressor genes like P53 and P21. ATR and ATM are upstream kinases that sense DNA damage and activate checkpoint kinase 1 and 2 respectively. ATR is primarily activated by replication stress or single-stranded damage to the DNA, while ATM responds to double-strand breaks. These kinases play a critical role in initiating the DNA damage response and activating the G2 checkpoint. Checkpoint kinase 1 and 2 are checkpoint kinases activated in response to DNA damage. They help stabilize the G2 checkpoint by phosphorylating and inactivating CDC25, thus preventing the activation of CDK1 cyclin B complex and delaying mitosis until DNA repair is complete. Checkpoint kinase 1, or CHK1, is particularly important for the DNA damage response, while CHK2 also responds to DNA damage, but has broader roles in the cell cycle. P53 plays an indirect role in the G2 checkpoint by regulating the expression of genes involved in the cell cycle arrest and DNA repair. When DNA damage is detected, P53 can induce the expression of P21, an inhibitor of CDK1, thereby reinforcing the G2 arrest. CDK1, also known as CDC2, is the primary CDK involved in the G2 to mitosis transition. It partners with cyclin B to form the CDK1 cyclin B complex, which is essential for initiating mitosis. The activity of CDK1 is tightly regulated by phosphorylation and dephosphorylation events, primarily involving the Wee1 kinase and CDC25 phosphatase. The Wee1 is a kinase that negatively regulates the G2 to mitosis transition by phosphorylating CDK1 on tyrosine 15, thereby inhibiting its activity. This phosphorylation prevents premature entry into mitosis, whereas CDC25 is a phosphatase that activates CDK1 by removing the inhibitory phosphate group added by Wee1. This dephosphorylation is a crucial step in promoting the G2 to M transition and the initiation of mitosis. Defects in G2 checkpoint regulators can lead to genomic instability and contribute to the development of cancer. For example, mutations in the P53 gene, which is involved in the DNA damage response, are common in many types of cancer. Understanding the regulation of the G2 checkpoint is also important for the development of cancer therapies, as targeting these pathways can sensitize cancer cells to DNA damage agents and improve the efficiency of treatments. Moving on to mitosis. Mitosis is a fundamental process of cell division that enables a single cell to divide and produce two genetically identical daughter cells. This process is crucial for growth development, and tissue repair in multicellular organisms. Mitosis ensures that each daughter cell receives an identical set of chromosomes, maintaining the genetic consistency across generations of cells. There are 4 phases of mitosis: prophase, metaphase, anaphase, and telophase. Back in high school, when I learned about mitosis, I came up with a mnemonic device to help me remember these phases in order. Please make another twin. Prophase, P. Metaphase, M. Anaphase, A. Telophase, T. Please make another twin. Moving forward, prophase. During prophase, the chromatin condenses into visible chromosomes. Each chromosome has already been replicated during the S phase of the cell cycle and consists of 2 sister chromatins joined at the centromere. The mitotic spindle, a structure made of microtubules, begins to form. The spindle fibers originate from the centrosomes, which are the microtubule organizing centers of the cell. Finally, the nuclear envelope, which encloses the nucleus, begins to break down, allowing the spindle fibers to access the chromosomes. During metaphase, the chromosomes align at the metaphase plate, with the sister chromatins oriented towards opposite poles of the cells. This alignment is crucial for ensuring that each daughter cell will receive 1 copy of each chromosome. The spindle fibers attach to the chromosomes at the kinetochore, a protein structure located at the centromere of each chromatid. The chromosomes start moving towards the cell's equatorial plane as the spindle fibers pull them. During anaphase, the cohesion between proteins holding the sister chromatids together are cleaved, allowing the chromatids to separate. The spindle fibers shorten, pulling the sister chromatids towards opposite ends of the poles, and separated chromatins, now individual chromosomes, move towards the poles of the cells. As the cell prepares for cytokinesis, the chromosomes reach the poles and begin to decondense back into the chromatin. Next, the nuclear envelope reforms around each set of chromosomes, creating 2 distinct nuclei within the cell. Finally, the mitotic spindle breaks down and the microtubules are reorganized to the next cycle cell. The M checkpoint, also known as the spindle assembly checkpoint or the SAC, is a critical control mechanism in the cell cycle that ensures accurate chromosome segregation during mitosis. The M checkpoint prevents the cell from progressing to anaphase until all chromosomes are properly aligned and attached to the spindle apparatus. The M checkpoint monitors the alignment of chromosomes at the metaphase plate or the equatorial plane of the cell during metaphase. For a cell to proceed to anaphase, each chromosome must be correctly attached to spindle fibers from opposite poles via structures called kinetochores, or protein complexes assembled on the centromeres of chromosomes, serving as attachment points for spindle microtubules. The checkpoint prevents the premature separation of sister chromatids by inhibiting the activity of the anaphase promoting complex cyclosome, or APCC, a key regulator of the cell cycle that triggers the transition from metaphase to anaphase. APCC, when activated, ubiquitinates and targets proteins like securin and cyclin B for degradation, allowing the separation of sister chromatids and progression to anaphase. The M checkpoint inhibits APCC until all chromosomes are correctly attached to the spindle. The M checkpoint is crucial for preventing aneuploidy, a condition where cells have an abnormal number of chromosomes. Aneuploidy can lead to developmental disorders and is a hallmark of many cancers. By ensuring accurate chromosome segregation, the M checkpoint helps maintain genomic stability across cell division. Key regulators of the M checkpoint include spindle assembly checkpoints like MAD1 and 2 proteins, BUB1 and 3 proteins, and the MPS1 protein. CDC14 complex, which activates the APCC complex, PDS1, and anaphase promoting complex. MAD proteins, or mitotic arrest deficient proteins, act as a scaffold protein that recruits MAD2 to unattach kinetochores. MAD1 forms a complex with MAD2 at kinetochores that have not yet attached to spindle fibers, helping to inhibit the progression to anaphase. This protein exists in two conformations, an open conformation, OMAD2, and a closed conformation, or CMAD2. When recruited to kinetochores by MAD1, MAD2 switches to its closed conformation, which binds and inhibits CDC20, a key activator of the anaphase promoting complex cyclosome, or APCC. This inhibition is crucial for the M checkpoint's function. Bub proteins are kinases that localize to kinetochores. They play a central role in the M checkpoint. BUB1 recruits other M checkpoint proteins, including BUBR1 and BUB3, which help to maintain the checkpoint signals. BUB1 also phosphorylates histone H2A, which is important for the recruitment of MAD1 and MAD2 kinetochores. BUB3 forms a complex with BUBR1 and CDC20. BUB3 helps to localize BUBR1 to the kinetochores and plays a role in inhibiting APCC by sequestering localized BUBR1 to the kinetochores and plays a role in inhibiting. I apologize. I'm losing my train of thought. BUB3 helps localize BUBR1 to the kinetochores and plays a role in inhibiting APCC by sequestering CDC20. BUBR1 functions as both a checkpoint protein and a pseudokinase. It forms a complex with BUB3 and inhibits CDC20, thereby preventing APCC activation. BUBR1 also plays a role in stabilizing kinetochore microtubule attachments. MPS1, a key kinase, is essential for the proper functioning of this checkpoint. MPS1 phosphorylates several of this checkpoint's components, including MAD1, MAD2, BUB1, which also enhances their activity and recruitment to kinetochores. MPS1 also amplifies the checkpoint signal in response to unattached kinetochores. PDS1, or precocious dissociation of sister chromatids 1, is a critical protein in yeast, particularly Saccharomyces cerevisiae, that plays an essential role in the spindle assembly checkpoint during mitosis. In higher eukaryotes, the functional equivalent of PDS1 is securin. PDS1 is stabilized by the mitotic checkpoint in response to unattached kinetochores or a lack of proper tension across sister chromatids. When the checkpoint is active, PDS1 is not degraded and separase remains inhibited, preventing anaphase onset. Cytokinesis, moving forward, is the process during the cell cycle where the cytoplasm of a single eukaryotic cell is divided into two daughter cells. The process typically occurs after mitosis and ensures that each daughter cell receives not only a nucleus, but also a roughly equal share of the cytoplasm, organelles, and other cellular components. Cytokinesis differs between animals and plants due to their structural differences. After mitosis, a contractile ring composed of actin filaments and myosin proteins forms just beneath the plasma membrane at the cell's equator, or the metaphase plate region. This contractile ring contracts, creating a cleavage furrow, which is a pinching of the cell's membrane. The furrow gradually deepens as the ring tightens. The two daughter cells completely separate. This results in two distinct cells, each with its own nucleus and a share of the cytoplasmic contents. In plant cells, cytokinesis cannot proceed through a cleavage furrow due to the rigid cell wall. Instead, vesicles from the Golgi apparatus gather at the center of the cell where the metaphase plate was located. These vesicles contain cell wall materials like cellulose. The vesicles fuse to form a cell plate, which grows outward towards the existing cell walls. As more vesicles fuse, the cell plate expands and eventually connects with the cell membrane. The cell plate matures into a new section of the cell wall that separates the two daughter cells. Once the cell wall is fully formed, the two daughter cells are physically and functionally separated. Cytokinesis is crucial because it ensures that each daughter cell receives a complete set of cellular components, enabling them to function properly and independently. Without proper cytokinesis, cells might end up with too much or too little cytoplasmic content or organelles, leading to dysfunction. Additionally, failures in cytokinesis can result in cells with multiple nuclei, a condition known as multinucleation, which can contribute to various diseases, including cancer. Oncogenes are genes that have the potential to cause cancer when mutated or abnormally expressed. They are often derived from normal genes called proto-oncogenes, which play essential roles in normal cell growth and division. When proto-oncogenes are mutated or overexpressed, they can become oncogenes and drive uncontrolled cell proliferation. Oncogenes can result from point mutations, gene amplification, or chromosomal translocations. These genetic changes lead to the production of proteins that either promote cell division excessively or inhibit normal cell death or apoptosis. The Ras proteins are a family of small GTPases that play a crucial role in regulating cell growth, differentiation, and survival. Ras activates the MAPK ERK pathway by interacting with a series of protein kinases. This pathway promotes cell cycle progression by stimulating the transcription of genes involved in cell division, such as cyclins and CDKs. Constant activation of Ras signaling can result in persistent activation of MAPK and P13K pathways, driving unchecked cell division and survival. The MYC or Mike gene is a family of regulatory genes that encode transcription factors crucial for cell growth, proliferation, and metabolism. MYC proteins function as transcription factors that bind to DNA and regulate the expression of a wide array of genes involved in the cell cycle progression, metabolism, and growth. MYC promotes the progression of cells through the cell cycle by upregulating genes that drive the cell division. Overexpression of MYC disrupts normal cellular homeostasis by driving uncontrolled cell growth and division. It can also lead to metabolic changes that support the cancer cell's rapid proliferation. HER2/neu signaling activates the cyclin D1 CDK4/6 complex. Cyclin D1 is upregulated as a result of HER2/neu signaling and is associated with CDK4 and 6 to phosphorylate the retinoblastoma protein. In many breast cancers, HER2/neu is overexpressed due to gene amplification. This overexpression leads to excessive signaling that promotes tumor growth, aggressiveness, and resistance to apoptosis. The BCR-ABL gene is a constitutively active tyrosine kinase, meaning it continuously activates downstream signaling pathways without the need for normal regulatory signals. BCR-ABL activates the P13K AKT signaling pathways. AKT promotes cell growth, survival, and growth by inhibiting the pro-apoptotic factors and stimulating cell cycle progression. These are the resources that I used.